Good evening and welcome. I'm Jean Johnson, one of the librarians at the Stanford Health Library. Thank you for joining us tonight for tonight's talk, Nutrition, Microbiome, and Pregnancy. It is my pleasure to introduce tonight's speakers. Christopher Gardner is the Rehnborg Farquhar Professor of Medicine at Stanford School of Medicine and the Director of Stanford Prevention Resource Center's Nutrition Studies Group. He's also the director of SPCR's Research Fellow Training Program. Justin Sonnenburg is an associate professor of microbiology and immunology at Stanford School of Medicine. And Scarlett Karakash is a clinical associate professor of obstetrics and gynecology, maternal and fetal medicine, and also the Director of Perinatal Services at Natividad Hospital at Stanford Children's Health. Throughout the program, please enter any questions that you may have in the box at the bottom of your screen. At the end of the talk, the panelists will try and answer as many of these as possible. All right, panelists, I turn it over to you.
Good evening, everyone. Thank you so much for joining us to discuss nutrition, the microbiome, and pregnancy, and also to talk about the study we've begun to learn more about how these three entities interact. My name is Scarlett Karakash. I'm a physician. I specialize in maternal fetal medicine, and essentially what that means is that I take care of women who have complicated pregnancies, and that could be due to a condition in the mom or a condition in the fetus. I am primarily a clinician. I'm really fortunate and I feel grateful to have teamed up with Christopher and Justin, who are experts in their field, so that we can learn more and collaborate to learn more about this topic. To start, I want to talk about why I think this topic of nutrition and the microbiome in pregnancy interests me from a clinical perspective. As an OB and MFM specialist, I usually have two patients at least. Oh, my slides aren't advancing. Okay. They are deeply interconnected mother-baby unit, and they influence each other's well-being. So in pregnancy, the mother can be affected with short-term health consequences and long-term, because certain pregnancy conditions have been associated with women's long-term susceptibility to disease, such as gestational diabetes or preeclampsia. And of course, the fetus is entirely dependent on the mother, and we know that the intrauterine environment, maternal conditions, and early exposures influence the fetus and the offspring's health. And that's really what we're going to focus on tonight, is the fetus and the neonate. So I wanted to start with the idea of the developmental origins of health and disease. And this is the idea that the intrauterine and early life environment has an impact on the health and the development of disease in offspring. Different pregnancy complications like fetal growth abnormalities, both large and small babies, preterm birth, maternal diabetes, hypertension, and obesity have been linked to shared or common long-term consequences to the offspring, such as hypertension, cardiovascular disease, diabetes, obesity, and metabolic syndrome. So we know some things about the developmental origins of health and disease, and more recently, we're getting more and more information about how the microbiome really plays a role in this. Studies suggest that the early microbiome has an important role in the development of the offspring's short and long-term health and immune development. An overview of potential mechanisms include microbial metabolite exposure in utero fetal colonization, which is controversial because traditionally we think of the in utero environment as sterile, but there have been some studies that challenge this theory. Also, different microbiome exposures at birth can all trigger fetal and neonatal immune responses, and this programs immune development. So some factors that shape the fetal and neonatal microbiome include the maternal microbiome, the gut microbiota, vaginal, and oral, as well as differing modes of delivery, such as vaginal delivery, cesarean birth, and then further on in development, exposure to breastfeeding or antibiotics, the environment, and a host of other exposures can really shape the infant and then the toddler's microbiome. What we are really focusing on is the maternal diet, because the maternal diet is really connected to the maternal microbiome. So the first question we want to know at baseline is, can the maternal diet and or the maternal microbiome influence the neonate's microbiome? And the answer is there have been human studies and many animal studies, but human studies that have shown that yes, the infant gut microbiome varies with the degree, for example, of maternal fat intake during pregnancy. Another study showed that fruit and dairy intake at certain points in pregnancy altered the clustering of infants into specific microbial compositions. And then in terms of the maternal microbiome's effect on the neonate's microbiome, they've shown that when they followed infant mother pairs for 4 months, the infants were transiently colonized by maternal skin, vaginal, and gut strains, and this persisted for up to 4 months. So we know that the maternal microbiome does play a role in the offspring's microbiome, and we know that this can be influenced by diet. So what are some of the associations that we have with alterations of the maternal microbiome and some adverse outcomes in the offspring? We know that a change in the maternal vaginal microbiome has been associated with preterm labor. Also, the gut microbiome changes have been associated with necrotizing enterocolitis, which is a serious gastrointestinal problem that affects mostly preterm babies and can be life-threatening. It's also been associated with adverse neurodevelopmental outcomes, metabolic disease, and obesity. I want to focus on preterm birth because for me as a clinician and obstetrician, this is something that really affects my patients. And in the US and worldwide, it's the #1 cause of infant mortality and morbidity. And infants who do survive often have long-term health problems such as cerebral palsy, intellectual disabilities, chronic lung disease, vision, and hearing loss, as well as a higher risk for other complications such as cardiovascular disease and hypertension. And the incidence is pretty high. It's 1 in 10 babies as of 2020 in the US are born preterm. And as you can see, we haven't made much of a dent in this. And when you break it up into racial and ethnic groups, Black women have a 14% risk of having a preterm baby. So this is really, really significant for us. And even after, despite many years of really intense research, the puzzle of preterm birth still has not been solved. And current interventions are really not very successful at treating or preventing preterm birth. And the title of this slide comes from a paper that was written by Dr. Romero in 2014 and sort of can illustrate why this may be. It's one syndrome with many causes. So we don't know all of the causes, and so it's very difficult to treat. But infection and inflammation is thought to play a huge role. Subclinical infection, which means the mom doesn't necessarily have a fever or abdominal tenderness or anything, but there is a small mild infection brewing. Intraamniotic infection, which is an overt infection, affects about 25% of preterm births. And then extrauterine maternal infections, such as a kidney infection or other reasons for sepsis or total body infection, is associated with preterm birth or can cause preterm birth. Microorganisms associated with preterm birth are thought to originate from the genitourinary tract and ascend through the cervix or through blood. Bacterial vaginosis is associated with preterm birth, and bacterial vaginosis is essentially the entity where the vaginal flora has shifted from primarily lactobacillus containing bacteria, which is considered normal, to an overgrowth of different other types of bacteria. And this is associated with preterm birth, but we know that treating BV does not prevent preterm birth. A little bit about the vaginal microbiome. So in normal pregnancy physiology, it seems that there is a lower microbiota diversity during pregnancy. It's usually dominated by lactobacillus, and we know that lactobacillus poor microbiomes have been linked to preterm birth. And the vaginal microbiome does differ by race and ethnicity, and there's still a lot we need to learn. So we talked about some factors that can influence the infant microbiome, such as maternal diet, mode of delivery, preterm birth, breastfeeding, and even if you have furry friends or pets at home, and other environmental exposures, and many, many more. And so when we think about the study that my colleagues Christopher and Justin will talk about a little bit more, there are some challenges when we try to study the microbiome in pregnancy and how diet can affect it, because there are a whole slew of conditions that can change or can affect the microbiome in the prenatal period. We have nausea and vomiting of pregnancy and food intolerance. We have gestational diabetes that can develop, preterm labor, and fetal complications. During labor, there's common use of antibiotics for various conditions, as well as differing modes of delivery. And in the neonatal and postpartum periods, women can either breastfeed or formula feed, and women and their babies may or may not receive antibiotics. So there are some challenges, but overall, I'm really excited about this study and what we can learn from it. And as far as I know, it's the first randomized controlled trial of dietary intervention in pregnancy, looking at the maternal and the infant microbiome. So it's exciting for me as a clinician, because I think down the road, we can learn a lot more that can help my patients. And so with that, I want to turn it over to Christopher, who's going to talk more about the diet and a lot more detail about what I was talking about right now. Thank you.
Fantastic. What a great opening. Thank you, Scarlett. I'm so happy to be here. I love the Stanford Health Library. And I'm going to continue with this slide set. Let me get it set up here. Thanks to Jean and Ryan and everybody listening in. So fabulous partners here. And I'm going to start this out by saying I've been at Stanford for almost 30 years, and I've done quite a few nutrition intervention studies in humans. In fact, I'm hoping somebody listening today was one of our participants in a past study. We've done quite a few of them. They tend to have a lot of generalizability to the population out there that's looking to try to change their diet to improve their health. And I couldn't have been more excited almost 10 years ago to meet Justin and Erica Sonnenburg, to add a whole new dimension to what I do, because my fun is playing with trying to get people to change their behaviors around diet, but there's always so many different things I can look at. And Justin's lab does some of the most exciting work in the nation around issues of the microbiome, health and disease. What are the preventions? What are the risks? What are the benefits? And he helped introduce me to the Human Immune Monitoring Center at Stanford, run by Mark Davis and Holden Maker. And so for the last many years, I have been feeding people and getting them to change their behavior, or at least my fabulous staff has. And then Justin's incredible crew has been characterizing the microbiome, and we've passed along some of our samples to Mark and Holden, and they've been looking at immune function, immune profiling. They've been looking at inflammation. And so we kind of have this nifty thing set up where we can pool all the resources that the three of us have together. But what's extra fun is the blank slate is there for us as to who do we work with and for what reason. And so it was really a thrill to meet Scarlett and say, what if we applied this to pregnant women? What if we were trying to look at the transmission of the maternal microbiome to the infant? And this is actually funded by a Stanford grad. She's on her third pregnancy. She's a total geek. She loves science. She loves metabolism. She loves the microbiome. And she was seeing that we really don't understand much about what happens in the terms of transmission of the maternal microbiome to the infant. So with some very generous funding, she is going to let us play with this. And I get the opportunity here to steal some of Justin's slides. So I need to tell you that these are his. And so there's going to be a little diet and microbiome lecture 101 embedded in our talk here, where he has pictured the small intestine being thinner here than the large intestine, the colon, and suggested that our human genome has about 20 genes that make the enzymes that will degrade carbohydrates. And the microbiome, the hundreds of species that live in our colon collectively, have over 10,000 genes to degrade fiber. So fiber is a type of carbohydrate, and the very definition of fiber is it's the type of carbohydrate that our 20 genes don't degrade. And so therefore, we don't break them down into small enough pieces to absorb in the circulation in the small intestine, and they all go down to the colon. So take, for example, a piece of cake, which is mostly just sugar and starch, and we actually have those enzymes. That's fine. So you've had a piece of cake with lots of carbs, and you've digested it all and pretty much absorbed it all. Here's some of the bacterial species in your colon. Do you see how sad they are? They're lethargic, they're lying on the side, they're depressed, they're not being fed, and they're hungry, they're ready for food. And on the other hand, if you were to eat broccoli, which would provide some carbs that you would eat, digest and absorb, but quite a bit of fiber. And the fiber would go to the colon. The bacteria would be happy. They have the enzymes to degrade them. They would break them down, use that fiber, those many different types of fiber for fuel, and generate small products, small metabolic products like short chain fatty acids that can be absorbed in our colon, go into the circulation. And we're observing that these likely have a positive effect on promoting immune function and lowering inflammation. Justin's term for this, for me, is a crack up, because he specifically is interested in the fibers that are accessible to the microbiota. So he calls them microbiota accessible carbohydrates. For short, that's MAC. So he talks about promoting a big MAC diet in the American public. Okay, sorry, that's a terrible joke, but it's great. I love to share it, but it's really not mine. It's his. So if you like it, it's mine. And if you don't like it, I don't know, actually it's his. And I think it's great. So the two different scenarios here are the complex carbohydrates that we eat or the simple carbohydrates that we eat. And they lead to very different situations here in the colon and for the microbiome. So this is one of the reasons why diet has a huge impact on our microbial diversity and all the metabolic products that they are using for fuel that we can then absorb in our gut. So fiber is an obvious one. So we've got veggies, we've got beans, we've got grains, we've got fruits. So one of the terms that we use for these overall is prebiotics. The pre meaning, so there are already bacteria in our gut, and this is the fuel that we're going to feed them. Now, I don't know how many in the audience have been hearing about pre and probiotics. I sure am in the mainstream media on blog posts and podcasts and magazine covers. And what I sense from talking to the general population is they not really sure if pre and probiotics are any different. So these prebiotics really refer specifically to fibers. And so there are lots of fibers, different fibers in food. There's different fibers in all those vegetables and fruits and grains and beans I just showed you. There's also Metamucil. There's also inulin, right? And so we've got commercial products that have fiber. In fact, there are a lot of products that have fiber. I, as a nutrition scientist, hope you will eat food, because food has lots of those fibers, but there are plenty in pills that you could buy. And there are a lot of studies going on right now where someone will focus on a single type of fiber and look at the microbiome. That is certainly one way to study it. It's not actually a way that we are going to choose to study it. And then the other major input that links diet to the microbiome is fermented foods, especially if the fermentation was done appropriately and the product is something that contains live bacteria. So if you're going to drink kombucha, eat plain yogurt, have kefir, sauerkraut or kimchi, all of these are fermented foods that contain live bacterial strains. If you've looked on the packaging, sometimes it'll say 20 billion CFUs, that's colony forming units. I'm pretty sure the low pH of your stomach acid kills off quite a few of them, but imagine eating 20 billion of them. Some of them survive and get to go down to your gut and may play a role in diversifying the microbial milieu that we have in our gut. Justin has just informed me that these slides I've been using for a couple of years are a little bit wrong. So I have always called these probiotics. Basically, the fermented foods contain bacteria that are similar to probiotics, but the key here is that the fermented foods have live bacteria. And if you did want to get them in a way other than foods, you could get them in pills. Here's one here with 40 billion CFUs. If you take this, I have to say, I'm a little skeptical with the commercialism that's going on here. I know that even in our vitamins and minerals, we don't really have good regulation from the FDA of what the content of those is. So nobody's really going around to verify that there's 50 billion or 40 billion or 30 billion as I'm looking around my screen. But it certainly is there's a strong commercial market for this, and these are appearing left and right. We decided to do a study recently using both of these. We got a group of 36 people to eat more fiber or more fermented food for 10 weeks. And we had to wrestle with, this is a research study coming out of Stanford. Wow, there's isolated fibers or there's multiple fibers. There's isolated bacteria and there's multiple bacteria in pills or foods. What should we do? Well, we really wanted to study food, and I'm super glad that Justin was on board for this. It's less controlled and rigorous than he might be able to do in a mouse study, which he's world famous for. But he's been anxious to work with humans. So we thought, wow, what type and how much? So I'll pose this to you all as citizen scientists. What do you guys think you would pick? Well, I'll tell you what we picked. We thought, there's so many different permutations. I'm not sure how we could get it right. Let's just ask people to eat fiber-rich foods, and it'll take all the different fibers they get. And some people might not like sauerkraut and like kimchi, or vice versa. Why don't we just try to get them to eat more fermented food, and we'll see what kind of signal we can get in the gut and in markers of inflammation. And then when it came out to how much, you might gasp in just a minute, but we were trying to get them to eat as much as humanly possible, hoping that, not to make this obscene, but we really wanted them to eat enough that if there was going to be a signal, we would see it. And you're probably all aware that changing human behavior around diet can be problematic, but it's not if you have Dahlia. Dahlia is our diet whisperer. She is this magician, super kind, super empathetic, patient and understanding. Another one of these total geeks who, as soon as she's done counseling our research participants, she goes and reads another research paper and another research paper so that she can be more and more informed. And in our study with these 36 people, we had them for 4 weeks each try to ramp up to get more and more fiber and more fermented food, and then maintain it for 6 weeks. And then we left them afterward they didn't have to eat none anymore, but we kept tracking them to see how much of a resilient or sustained response they might have. And Justin's lab, which I will not cover, did all the measurements to see what kind of impact we might have. And so I'm really proud of my nutrition group that I get to work with, an incredible set of staff. I'm going to quickly walk you through what we did. We wanted the fiber folks who are typically getting on average about 20 grams of fiber a day to eat more. And just focusing on the left for a minute, we got them to get up in this ramp phase to over 40 grams a day. So we got them to double their fiber. Okay, now step over to the right for just a minute. Folks in our study were almost eating no fermented foods. We said, how much can you eat? Look at this, 2 doses a day, 4 servings a day. We got them up around 5 servings a day. Now, that might sound ridiculous, but I do want to point out that a half a cup of kimchi or a half a cup of sauerkraut is a serving. And if you get those bottles of kombucha in a store, those are 2 servings. So if somebody were to have a cup of sauerkraut, drink a bottle of kombucha during the day, and then have a cup of kimchi at night, that would have been 6 servings. And actually, if you put it into terms of calories, that was only about 300 calories. So most of their calories were not affected over the course of the day, but we did get them to double their fiber, go from almost 0 to 5 or 6 servings a day of fermented food, after which they maintained them. The study was over right here. I guess we're a little excited that by introducing them to these fiber-rich foods and these fermented foods, some people actually maintained those behaviors. We're super excited that Justin's lab did such a great job with this. He has some fabulous postdocs and PhD students. We got this published in Cell. There's something called an altmetric score, which is sort of how much visibility and press your article gets. This is actually an incredibly high visibility. We were in the New York Times once for the study, and then there was so much interest in it afterward, they followed up and said, this is how to get it in your diet. So we were in the Sunday New York Times, Tuesday Science section, 2 weeks in a row, looking at the impact of this high fiber diet and high fermented food. So moving forward, Justin is now going to tell you more about what he finds in the microbiome and what he finds when we work with the Human Immune Monitoring Center. And we're all going to pair together and do this in pregnant women and their infants. So turning it over to Justin.
Awesome. Thank you, Christopher and Scarlett, for the wonderful talk to this point. How great is this that I get to work with these two? I mean, what a dream come true. So it's wonderful to be here with all of you. I wanted to start off with just this image of the gut microbiota in situ. It's a image that was taken by one of my former graduate students. And this is a cross-section of colon. And everything you see in the top left there is the gut microbiota. So each one of those colorful little rods is a bacterial cell, and you can see how incredibly dense this community of microbes is. And that is essentially fecal matter. So this is how photogenic fecal matter can be if you treat it the right way and capture it at the right angle. What is amazing about this picture to me is not only how dense this community of microbes is, but how close it is to our tissue. So in this cross section, this is the lumen, what we call the lumen of the gut. The colonic tissue is down here on the bottom right, the blue area, and then there's this green stripe running through the middle, which is the mucus layer, and that's what we secrete out of our cells to keep the microbes in the right spot. And so if that mucus layer goes missing, these microbes get too close to our tissue and start to incite inflammation. But as long as we've all heard the phrase, good fences make good neighbors, that's true also in the gut. The mucus really keeps these microbes in the right spot so that they can help us with all the things that they help us with. And so what an incredible interface for exchange of information. There's all kinds of flow of chemicals between the microbiota and the host, incredibly dynamic environment. And we know that this is an incredibly complex community. We harbor trillions of microbial cells. They're actually not just in our gut, they're all over our body, but most of the microbial cells associated with our bodies occur in our colon, in the far reaches of our digestive tract. And these microbes, there's on the order of hundreds to thousands of species, depending upon how you define a bacterial species. But the composite genome greatly eclipses the coding potential of our own human genome. So we know that the combined microbial genome is approximately 100 times or more in size than our human genome. So incredible coding potential. Most of these genes, we don't even know what their functions are. So this is a big challenge for the field to establish all of this. And then a major question is not just who is there, which microbes are living inside of us, but also what are these microbes doing? How do they integrate with our biology? And so when you take this in whole, I think, the really important thing here is that we aren't just one species. Each one of us, each human, we think of as a human species, one species. We're actually an ecosystem composed of hundreds to thousands of species. And it's really important that all these species work in concert one of these species being Homo sapiens, for us to have optimal health. And you know, we've known for a long time that the gut microbiota is really important in helping us digest food, in gut health. But what's been really incredible over the past decade or so is that the incredible field of research, microbiome science, has unfolded and told us that gut microbes are actually involved in essentially all of our biology, that they're involved in dictating aspects of our metabolism, our immune status, and there's even the brain-gut axis, so impacting our central nervous system. And the fact that our gut microbes are malleable, we can change the composition and function of this community, individualized, and also connected to so much of our biology, it really is clear that the gut microbiome will be an important part of precision health going forward. And so we like to think about what are the set of tools and knowledge base that we need to develop that will allow us to shape these microbes in a way that gives us improved health, that can help to treat and prevent disease. And so there are a variety of different approaches for this. Many of you may have heard of fecal transplants, the idea that you can essentially reboot a diseased microbiota with a fecal sample from another individual, basically just introduce healthy microbes. And this actually works very well to cure recurrent Clostridium difficile colitis. Other diseases, it's not clear how effective, it's probably not as effective, but it does bring up the question of what are the limits of using the gut microbiome to exact change on our health. And so while there are a variety of approaches to impacting our gut microbiome, one of the major themes in the field, and certainly something that we're very interested in, is the role of diet in shaping the gut microbiome in health and disease. And so one of the things that's become very clear in the field as we've studied different populations around the world is that individuals like all of us living in the industrialized world have a gut microbiome that is losing diversity. And it's also becoming clear that when it comes to the gut microbiome in the industrialized world, higher diversity corresponds to better health. And so we're really interested in thinking about how do you reconstitute a more diverse, healthy gut microbiome. And as it is right now, we're headed towards something like this clear-cut forest. And this is likely due to a variety of factors associated with our lifestyles, things like antibiotics, excessive sanitation, but certainly diet plays a really important role in dictating the microbes and what they're doing in our gut. So we know that as Scarlett mentioned early in the talk, the microbiota is passed down through generations. We know that the microbes that a baby receives will be affected by whether that baby is born vaginally or born by C-section, whether that baby is breastfed versus formula fed, whether there are pets in the household, whether that baby receives antibiotics throughout their life. And so there's really a huge number of factors that shape the gut microbiome. And we know that our gut microbes dictate aspects of our immune status, aspects of our metabolism. And it really begs the question of whether we can change the trajectory of infants in a positive way so that they're set on a positive trajectory for better health with a more diverse gut microbiome. It's important to also recognize at this point that all of this implies that your diet not only influences your microbiota, but it also influences the microbiota of your progeny. And so, a major question that we've really thought a lot about is, can we repair our gut microbiota and improve our health? And one thing I'll note here is that many of us in the field use the terms microbiota and microbiome interchangeably. So you'll see both of those terms and you'll hear me use those interchangeably. So can we repair this gut community, the gut microbiota, to improve our health? We know that this low diversity microbiota is associated with the development of many chronic diseases, metabolic diseases, inflammatory autoimmune diseases. And so can we, if we change our lifestyle, change our diet, can we improve our gut microbiome? And so, we originally, when we came to Stanford, studied mice to try to answer some of these questions in a very controlled, mechanistic experimental environment. And then rapidly came to see that we, were interested in diet. We wanted to study diet, and it made sense to actually do dietary experiments in humans, because diet is not considered a drug, and you can basically do dietary interventions in humans and look at the impact. But of course, we're terrified of humans. Humans are terrifying. And so luckily we met up with Christopher, whose group specializes in doing dietary interventions in humans. It's been a really marvelous collaboration. We have a variety of other supporters who have been integral to the development of the Center for Human Microbiome Studies that we started: Mark Davis, Kari Nadeau, David Relman, and others. And Erica Sonnenburg, my wife, leads the laboratory with me. So we've all been working on this idea that the new trajectory of research should not focus on our traditional paradigm of working on molecular mechanisms, working through animal models, and then trying to translate into humans, but instead doing dietary experiments in humans, as Christopher described to you previously with our fiber fermented food study, and then using those findings to reverse translate back into animal models to try to establish what the molecular mechanisms and causal relationships are. And so you've seen this slide. This is just to remind you that simple carbs starve your gut microbiota. If you're eating, just protein, fat, simple sugars, starch, all of those nutrients get absorbed in your small intestine. That leaves your gut microbes starving. If you're eating a plant-based diet with a lot of fiber, that provides good sustenance to your gut microbes, because most of them live at the far reaches of your gut. They don't get first crack at all these really high prized nutrients. So you have to be eating dietary fiber to properly feed your gut microbiota. If you don't eat a high fiber diet, your gut microbes will actually start eating you. That green mucus layer that lines the gut is actually made of carbohydrate, and it's a backup food source for your gut microbes. So if you're not eating fiber, we know that gut microbes turn to start eating the mucus layer. They can deplete that mucus layer and start to incite inflammation. So dietary fiber, we think, is really important. We also know, especially in the face of a pandemic, that a sanitized environment is extremely important for good health and to not transmit germs, but it may also have a cost in preventing us from sharing healthy gut microbes back and forth. So one of these, approaches that minimizes infectious disease may have negative consequences for our gut microbes. Also the case with antibiotics, really important medications in the right context, but certainly a lot of collateral damage for our gut microbes. And so we also know that humans and microbes have a long history together. This is an image of a hunter-gatherer in Tanzania, hunter-gatherer from the Hadza population. This is a person who actually has washed their hands in the digestive contents of an animal that they've slaughtered. To get the blood off of his hands, he actually just rubbed his hands in the digestive contents. And so you can imagine, the difference of their microbial exposure in Hadza land compared to those of us that live in the United States. We also know that fermented foods are living foods and have a lot of microbes in them. And perhaps one safe way of reintroducing microbes and environmental microbial exposure back into our lives is to eat fermented foods. And so with this study that Christopher mentioned, we really address, can high fiber foods or fermented foods improve the microbiota and improve health? And so he's introduced this study design. This was just an amazing study to watch an incredible team execute. Christopher's already mentioned Dahlia, really the key to getting people, the free living humans, to eat differently and to be well educated about what they're eating. We also had wonderful trainees, Maddie, Hannah, and Gabby, that are just among the many people that played a really critical role in this study. But the goal really was just to monitor in the blood, can we change the immune system through comprehensive immune profiling of these individuals, at the same time monitor their stool and look at what we're doing to the microbiome with either a high fiber or high fermented food diet. And so the high fiber diet consists of course, legumes, whole grains, vegetables, fruits, nuts, and so forth. The high fermented food diet, kombucha, yogurt, kefir, kimchi, sauerkraut. And so we went in and looked at these. So this is just to remind you that, Christopher's group was incredibly successful at getting people to eat a lot of fiber and eat a lot of fermented food. And the question is, what happened to their gut microbiome? So we surveyed the gut microbiome. These are plots of the group's diversity over time, over the course of the study, for the high fiber group and the high fermented food group. And what this green chart on the left is showing us is that the number of species started off just, a little over 100 at baseline and didn't change that much over the course of the study. And this was very startling to us that we weren't able to increase gut microbiota diversity with this beautiful high fiber diet. However, the high fermented food diet did increase diversity. This group of people statistically increased their gut microbiota diversity over the course of the study. Now, this may not look so impressive, but in the context of all the human studies that we have been seeing coming out in the literature, this is quite a profound change to gut microbiota diversity over such a short time period. And so the question is, what happened to the immune system? Did the immune system change its status over the course of the study? And what we see is in the fermented food cohort, there was a tremendous decrease in a number of inflammatory cytokines. Each one of these plots shows a different blood marker of immune inflammatory state. We see that, for instance, famous ones, interleukin 12 decreases, interleukin 6 decreases. So several markers of inflammation decrease as these people are eating fermented food and increasing their gut microbiota diversity. We also used other parameters of immune status. This is a technology called CyTOF, looking at immune cell signaling. We can see that a number of different cell types, monocytes, CD8 T cells, CD4 T cells, and B cells, mainly decrease the phosphorylation status, the activation status of many signaling pathways, indicating decreased inflammatory status as well. So really a broad signature of decreased inflammatory status over the course of this intervention. Now, the question is, why did the fiber intake not result in substantial changes to the microbiota or immune status? And there's a little more nuanced. I won't get into the details since we don't have time, but we think that the depleted gut microbiota doesn't have the right microbes to degrade the dietary fiber, and it's passing through the industrialized microbiome, unfermented, somewhat inert. And so it brings up the question of, if we combine high fiber and high fermented foods, could we improve the microbiota and health of the mother and the baby? And this is what we're embarking on now. And I'll hand it back to, I don't know if Christopher or Scarlett, you want to take it over at this point.
So this is what we've had a tremendous amount of fun designing with Scarlett, Justin, a bunch of other colleagues. We would like to find about 130 women who are pregnant that would go into 4 groups. One would mirror our past high fiber diet group. One would mirror the high fermented food group. And one of the things that we didn't have really time to show you is that the fermented food group not only saw a number of species appear, but most of them were not from the fermented foods we were giving them. About 95% of them seem to have perhaps been below the level of detection before. And then when we changed the environment of the gut, they appeared. So maybe we would do a better job digesting that fiber if we could increase the diversity of the microbiome first. So we basically have an A, a B, an A plus B, and a control group. We're going to try to get these women to participate during their pregnancy, before birth, come in a few times, give us stool and blood samples. Then we'll capture everybody at birth and follow them for 12 months, probably 18. Maybe if the donor is really happy with this, we might actually try to follow the kids even farther than that. And it's, as is typical of Stanford, we just have an incredible array of tools to use. So this is probably too much detail for you, but we want to get too excited about this so that you will tell any friends, any daughters, any cousins, any nieces who are pregnant about the opportunity to sign up for this study and learn a lot about mom's microbiome, the transmission to the infant, and the infant microbiome, which requires some either stool samples from the mom or diaper samples from the kid. We've got different ways to draw blood. We're going to take vaginal swabs and breast milk. We're going to ask about the diet. We're going to capture all this diet all the data here. We might get some cord blood at the time of birth. We might get a drop of blood from a heel stick. But this is a combination of mom and infant during pregnancy, and then for 18 months, 12 months, probably even longer, and actually our study coordinator came up with this name, the Maternal Offspring Microbiome Study. Isn't that great? The mom's study. So FI-FO is for fermented food, fiber, and food. Fermented fiber food for moms. Love the acronym. Hope that's easy to remember. We're going to make this accessible so that it can be passed out to folks so that you could share it with, again, daughters, nieces, friends, neighbors. A set of instructions of how you find out if you're eligible, a QR code if any of you are up on that. If not, we have some links that they could go to. And that is what we wanted to present to you today. We know some things. There's actually more things we don't know than we know. We have taken advantage of this trifecta of fabulous colleagues that are all around the corner from one another at Stanford. And we are going to be ready to take some of your questions now. We hope we generated some. We have at least 10 minutes for questions. I guess we should be looking at the Q&A. So Justin and Scarlett, there is one about autism in there from the very start of the talk. I don't know how many, some of you are using the chat. I guess we can try to look at both. So participants, can you see that there is both a Q&A and a chat option?
Yeah, and I actually typed a quick response earlier to the autism question. I think
okay, great.
To follow up on that, this is a field unto itself. We don't study this. I'm familiar with some of the literature. Very interesting connection between gut microbiome dysbiosis and autism. There are mouse models of this where investigators, particularly Sarkis Mazmanian and Elaine Shao down at Caltech and UCLA, have looked at this to try to understand causality and have shown relationships between leaky gut, microbial metabolites getting across the gut barrier, leading to exacerbation of a mouse model of autism. But it's a big field that's rapidly expanding, and I think much like many other diseases that have been associated with gut microbiome dysbiosis, it's really hard to know the causality, which way the arrows point. Is the gut microbiome dysbiosis a cause of the disease happening in parallel? Is it causing the disease, potentiating it? So very interesting field. Unfortunately, not a lot of answers that I'm aware of yet.
Another one of our questions is super relevant to us. How do I find out about the study? I'm 6 weeks along. So we will be sure to share the directions. We will ask the Stanford Health Library to pass that out. So that QR code I had up on the slides. Actually, I think you guys are all going to get access to the recording of this. And so listener who asked that question just now, if you want to go through the slides, the QR code will be part of the slides. And if you're not familiar with QR codes, there's an email in there, something that you can click on. Worst comes to worst, send it to cgardner@stanford.edu and I'll hook you up, or even nutrition.stanford.edu would work. And we've got another one about how quickly some of this happens. Justin knows how quickly this happens. We've been working with the plasticity of the gut. And Justin, are you seeing that in the chat?
Yeah, I was actually trying to dig up the QR code to put that in the chat. Let me hang on 1 second.
Oh, thanks.
I can.
So while he's digging it up, I can tell you that we have been wrestling with how quickly this happens. We, in most of our studies, have designed them to be 10 weeks long, 6 weeks long, something like that. But we've been collecting samples every 2 weeks. We don't actually have all the funds to go back and look at every sample every 2 weeks, but it certainly can change in 6 weeks. We have some of the samples on ice where we're trying to see if it would change faster than that. To add a different twist to that, I don't know if Justin will want to speak to this. We have actually published another paper that came, a secondary analysis from a weight loss study that we had done, where we had collected data from 600 people who lost weight over a year. And we had samples at 3 months, 6 months, and 12 months. And we saw some interesting difference between our two study diets at 6 months that were gone at 12 months. It's a little hard to figure out if the disappearance of the difference was because folks had tended back toward their pre-study diet, or if there was some resilience to the microbiome that after changing, it had shifted back to its original pattern. So maybe I'll throw that one toward Justin. He's done with that link.
Yeah, I couldn't find the QR code, but I did find a URL that I'll put in the chat and in the Q&A. Yeah, Christopher, your point's an excellent one. That I think the changes to the gut microbiome can happen really rapidly if you have a really profound perturbation like antibiotic treatment. For dietary changes, quite often what's been reported is rapid changes with sudden changes to diet, but then a gradual return to something close to the baseline state. So this incredible resilience of the gut microbiome to change, which is why the finding in the fermented food study was such an interesting one, because that such a change in diversity is not seen to persist in studies. Quite typically, if you see a change in diversity, it'll usually go back fairly quickly. So so changes can happen quickly, but there's an incredible stability to the gut microbiome in the absence of something monumental like diarrhea or antibiotic treatment. The community is typically pretty resilient to change.
There are a couple of questions in the chat about the dietary fiber. Maybe for you, Christopher, can you explain again how you're increasing dietary fiber? And also another question about does over-the-counter fiber supplement count?
Sure, happy to take that on. Yeah, there are so many fibers that it is difficult. We actually do hope, so maybe Justin will agree. So this idea of precision nutrition could be that we characterize the microbiome. We see there's an important species missing or in low prevalence. We've identified that really wheat berries are the best source of that fiber. We're suggesting you introduce that, or this particular brand of kimchi is going to introduce that bacterial strain. We're not there yet. We really don't know what that is. And a lot of these companies that are offering to have you send in your stool samples, and then they tell you, oh, yes, you should be a forager. You should be a high protein eater. You should be a Wild West something. They don't really know that. So what we really need to be doing here is collecting those data to see to what extent we can link specific fiber or specific species to some of these things. So our approach for the moment, this doesn't sound very risky. Eat a healthy diet. Eat a healthy diet with lots of veggies, beans, whole grains, and fruits. And that way you will get a variety of fibers, which might in the end be one of the best answers. So that's the approach we're taking so far.
Yeah, and I agree. Let me just add one thing that, there have been a couple studies looking at purified fibers in dietary studies similar to the one we did, but not monitoring the immune system, just monitoring the microbiome. And if you consume a single purified fiber at a high dose, the studies that I've seen, gut microbiota diversity drops because you end up with one or two species that are really good at using that specific dietary fiber blooming to high levels, taking advantage of that single fiber. And so what Christopher is saying is supported by that if you go to a salad bar and you load up on a bunch of different types of plants with a huge diversity of different types of dietary fiber, you're more likely to encourage a more broad diversity in your gut microbiota, I think.
And then for the sake of taking a quick question, one is about soluble versus insoluble fibers. So the soluble fibers are more accessible to the bacteria than the insoluble fibers. Dustin, correct me if I'm wrong, but the almost moot point is, if you've ever seen a graphic of all the foods that have fiber and how much soluble versus insoluble fiber there is, most plant foods have both. It would be really hard for you to say, oh, I've heard insoluble fiber is bad and soluble is good, so I'm only going to eat soluble fiber today. I'm going to avoid all insoluble fiber. We don't actually have those foods. You couldn't do that. So there probably is a high preference for soluble, correct, Justin? For the
Yeah,
bacteria?
I think that's right. And there's some evidence that having a slow fermentation along the length of the colon, as opposed to this big burst, like if you're eating a lot of soluble fiber, it's probably pretty easily accessed by microbes and rapidly fermented, and therefore could be rapidly fermented in the more proximal portion of your colon, which can lead to both discomfort, but also may bypass some of the benefit of slow fermentation that happens over the length of the colon as the contents is moving through it. So there's probably good utility to both soluble and or range from soluble to insoluble, like you'd get in whole plant-based foods. And then, one question that before it scrolls off here, I see a question: are you seeing continual increase in biodiversity of the participants that keep up the fermented food diet? We haven't looked beyond the time points that we've shown, and so we're excited for the FIFO mom study, where we'll actually have the intervention proceeding for a longer period of time, and hopefully we'll be able to see if biodiversity plateaus at some point or if it keeps on going up. It's a good question.
I just typed an answer. Scarlett's working on typing an answer.
Is there a risk in eating fermented foods while pregnant? Heard no kombucha because not pasteurized and so potential for food poisoning.
Yeah, so we're going to, avoid kombucha in pregnancy because of that, and also because of the small amount of alcohol. And other than that, there is no known increased risk for fermented foods in pregnancies, and a couple of studies have looked at this specifically, and there's no risk.
So everybody, I think we're at the end of our hour. We want to be respectful of your time. I think Jean is going to close up for us. I just want to give a huge shout out to the Stanford Health Library, to Stanford as a whole, and in particular to my amazing colleagues.
Well, thank you, doctors, for sharing this great information. Thanks also to our audience for joining us tonight and for the really good questions, I thought. If you'd like additional resources and information, you can contact the Stanford Health Library. And we hope you'll join us for our lectures in the future. All right, good night.
Thanks, everyone.
Good night, everybody.
Bye-bye.